You send saliva in a tube, wait 6 weeks, open an email with percentages, 32% this, 18% that, 4.7% something you've never heard of. You see ancestors mapped across continents, migration routes sketched in confident lines. Your genetic story, they say. Complete, verified, scientific. Now forget everything you think you know about what DNA testing can actually tell you. Because in 2003, something disappeared from the Human Genome Project's final report. Not a mistake, not an oversight. A deliberate removal of genetic signatures that didn't match any known population on Earth. Sequences that suggested something impossible. A human lineage with no modern descendants. Or rather, descendants who've been systematically reclassified, redistributed, and erased from every commercial DNA database you can access today. This is about why certain bloodlines don't show up in your results, why specific genetic markers vanished from reference databases, why no ancestry company will tell you if you carry sequences that match architectural sites they won't let you visit, why your DNA test won't show Tartarian blood, and what they deleted from genetics to make sure it never would. I started in a place I didn't expect, not a library, not an archive, but the customer service response to a simple question. It was late 2019. I'd taken one of those popular DNA tests, the kind advertised during holidays, the kind that promises to reveal your ethnic makeup with scientific precision. My results came back typical: British, German, Scandinavian, fractions of Eastern European. The usual European mixture you'd expect from someone with my background. But I'd noticed something strange while reading about genetic haplogroups. Those deep ancestral lineages, traced through specific genetic markers. There were references in older papers to haplogroups that no longer appeared in any current database. Not marked as rare, not listed as extinct, simply absent, removed from classification systems entirely. I sent a question to the testing company's customer service. Simple, direct. Why are certain haplogroups from pre-2004 papers no longer listed in your reference populations? The response took 3 weeks. When it came, it was 6 sentences long. Our reference databases are updated regularly to reflect current scientific consensus. Older classification systems contained errors that have since been corrected. We use only validated population groups in our ancestry reports. Thank you for your interest in genetic genealogy. That word, errors. I teach biology at a university. I've read hundreds of genetic papers. When scientists discover classification errors, they publish corrections. They explain what was wrong, why it was wrong, how they fixed it. The literature becomes clearer, more accurate. What they don't do is delete entire haplogroups without explanation. What they don't do is remove reference populations from databases and then refuse to specify what was incorrect about them. So I went looking for those errors. The deeper I looked, the more I found not corrections but absences. Papers from the late 90s, citing haplogroups that don't exist in modern databases. Population genetics studies from 2001, 2002 referencing genetic signatures that commercial testing companies no longer include in their algorithms. And everywhere, the same phrasing, updated to reflect current consensus, reclassified according to revised standards, removed due to insufficient verification. Never. We were wrong about this marker for this reason. Always. The polite language of erasure. I couldn't unsee it. Every genetic database I checked told the same story through its silences. Haplogroups present in 2002, gone by 2005. Reference populations cited in academic papers, absent from commercial testing panels, and the timing centered tightly around 2003, the year the Human Genome Project published its completion, the year they declared they'd mapped all human genetic diversity, the year they stopped looking for sequences that didn't fit. The International HapMap Project headquarters sits in a building you'd walk past without noticing. Gray concrete, small windows. The kind of institutional architecture designed to be forgotten. I visited in early 2020, before travel became difficult, expecting to access their archived genetic databases through their public research portal. The woman at the information desk was polite, professional. She pulled up my request on her screen, scrolled through the database access form I'd submitted weeks earlier, and then looked up with an expression I was starting to recognize, the carefully maintained neutrality of someone delivering pre-scripted responses. I'm sorry. Access to pre-2004 raw data files requires institutional review board approval and must be requested through your university's genetics department. I have IRB approval, I said, sliding the document across her desk. From my university, for comparative population genetics research. She glanced at it, didn't pick it up. You'll need specific authorization from the data custodians. Each dataset has individual access requirements. Which custodians? That information is in the database. The database I can't access. She smiled with practiced patience. Exactly. I asked if I could speak with someone from the archival genetics team. She made a phone call. 20 minutes later, a man in his 60s appeared, research coordinator, he said, though he never gave his name. We talked in the lobby. He wouldn't take me upstairs. I asked about haplogroups listed in papers from 2000, 2002 that no longer appeared in current databases. He nodded as if expecting the question. The early HapMap data contained provisional classifications that were later refined. Some turned out to be sequencing artifacts. Others were reclassified into existing groups when more samples became available. Can I see the reclassification documentation? It's in the published updates. I've read the updates. They don't specify which provisional groups were artifacts and which were reclassified. They just list the current validated haplogroups. He looked at me with something like sympathy, or maybe resignation. The thing you need to understand about the Human Genome Project is that it created standards. Before 2003, genetic classification was chaotic. Different labs using different markers, different naming conventions, different reference populations. The completion of the project meant finally having consistent, verified standards. What happened wasn't deletion, it was standardization. Standardization that removed entire population groups from reference databases. Standardization that removed provisional categories that couldn't be validated. Why can't I access the raw data to see which samples were invalidated, and why? His expression didn't change. Privacy protections. The samples came from individuals. Even anonymized genetic data requires authorization. From 2001, from any year. Genetic privacy is retroactive. I asked about a specific haplogroup, one I'd found referenced in 7 different papers between 1999 and 2002, all studying populations in Central Asia. The papers called it R2b. M207 described it as geographically restricted to the Altai region with no apparent relationship to surrounding populations, noted its unusual combination of markers suggesting ancient isolation. By 2005, our 2% didn't exist, not reclassified, not marked extinct, just gone. Do you know that haplogroup designation? I asked. He was quiet for several seconds. Then, early classification systems often created overly specific categories based on limited samples. When more samples became available, they fit into broader established groups. Which group did Dar2bog, M207 fit into? I'd have to check the reclassification records. Can you? Not without data access authorization. I left Geneva with no data, no authorization, and a growing list of genetic sequences that officially never existed. But I also left with something else, the man's hesitation before answering, the careful way he chose words, the refusal to say directly that those sequences were errors, the way you'd speak if you were describing something real that you'd been instructed not to acknowledge. The Institute of Genetics and Physiology sits at the edge of the city, where Soviet-era buildings give way to newer construction. I met Dr. Sol Bektarova in her office on the 3rd floor, windows facing the mountains, shelves packed with binders, a computer displaying scattered genetic sequences. She'd published 3 papers between 2000 and 2002 on population genetics of the Altai region. Her work had identified unusual genetic markers in isolated populations near the Kazakhstan-Russia-Mongolia border, markers that suggested either ancient migration patterns no one had documented, or as she'd carefully phrased it, potential archaic admixture from an unknown source. Her last paper on the subject came out in late 2002. After that, nothing. Her publication record shifted entirely to agricultural genetics, plant breeding, crop optimization. 15 years of human population genetics research, abandoned. I asked her why. She was quiet for a long time. Outside her window, the Tian Shan Mountains caught afternoon light. Finally, she said, "You read my 2002 paper? All 3 papers. You found genetic signatures in Altai populations that didn't match any known reference groups. You suggested they might represent an isolated lineage, possibly dating back thousands of years. I suggested that in my draft feed." She pulled a binder from her shelf, opened it to a page dense with highlighted text. "This is what I submitted for publication. This," she turned several pages, "is what was actually published. You see the difference." The submitted draft included detailed analysis of specific genetic markers, including the R2ba M207 sequence. It described how these markers appeared in 37 individuals from 3 different villages, always in combination with other unusual sequences, never appearing isolated. It noted that the pattern suggested a coherent genetic lineage, distinct from all surrounding populations, possibly preserved through geographic isolation over millennia. The published version removed all of that. It mentioned genetic diversity consistent with historical isolation and markers of interest requiring further study. The specific sequences weren't listed. The R2b M207 designation didn't appear. The suggestion of a distinct lineage was gone. The editor said the claims were too strong for the data, Dr. Bektarova told me. Said I needed larger sample sizes, more validation, comparison with additional reference populations, all reasonable concerns. So I collected more samples, expanded the study to 5 more villages, found the same markers in 42 more individuals. And I submitted the expanded study in early 2003. It was rejected, not for methodological reasons. The rejection letter said, "I still have it." She pulled out another document. "The genetic markers described in your manuscript do not correspond to any validated haplogroups in current reference databases. Without proper classification framework, publication would introduce confusion into the literature." I read the letter. It was dated March 2003. "They rejected your paper because you found something not in the database. They rejected it because I found something they were removing from the database." She pulled up a file on her computer, genetic sequences, raw data, allele frequencies. "This is from my samples. This," she pulled up another file, "is from the HopMap reference database, accessed in 2001. You see this marker combination? I saw it. A specific pattern of single nucleotide polymorphisms, a genetic signature as distinctive as a fingerprint. Now look at the same database region, accessed in 2005. She pulled up a third file. The marker combination was gone, not marked as invalid, not listed under a different classification. The reference sequences simply skipped over that genomic region, as if those particular positions had never been examined. I contacted the HapMap consortium," Dr. Bektarova said, "asked why reference samples containing these markers had been removed. They said those samples failed quality control checks. I asked what specific checks they failed. They said the information was in their quality control documentation. I requested the documentation. They said it was available to authorized researchers through their data portal. Let me guess, you couldn't get authorization. I got authorization. I'm a published geneticist at a recognized institution. They gave me access to the data portal. But the quality control documentation for samples removed between 2002 and 2004 isn't there. There's a section labeled Deprecated Samples, see Archive, but the archive link returns an error, has for 15 years. She closed the files, looked at me directly. I stopped publishing on human genetics because there was no point. Every paper I submitted that mentioned these markers was rejected, not for bad methodology, not for insufficient data, for finding genetic signatures that no longer officially exist. Eventually I realized I could keep fighting a system that wouldn't acknowledge what I'd found, or I could work on something else. So I switched to plants. Plants don't have genetic sequences that databases delete. I asked if she still had her samples, the blood samples from those 42 individuals in Altai villages. I have the extracted DNA, stored at -80 Celsius. I couldn't bring myself to destroy it. She paused, "But I can't test it anymore, at least not for these markers. The primers you'd need, the specific genetic probes to identify these sequences. Commercial companies stopped manufacturing them after 2004. When I tried to order them in 2006, I was told those primer designs were no longer supported. No explanation, just discontinued. Can you synthesize custom primers? Technically yes, but you need the sequence data to design them. And the sequence data for these markers has been removed from every public database I can access." She looked at her computer screen, at the files containing data that officially doesn't exist. "I have genetic samples that I can't fully analyze because the tools to analyze them have been deliberately made unavailable. Do you know how that feels? To hold physical evidence you can't examine because someone decided the examination shouldn't be possible." Before I left, Dr. Bektarova gave me a list. 7 other researchers, she said, people who'd published papers between 1999 and 2003 about unusual genetic signatures in Central Asian, Siberian, or Eastern European populations. They all stopped publishing on human genetics around 2003 or 2004, she said. Some switched fields like I did. Others just stopped publishing at all. I looked at the names, recognized 3 of them from papers I'd already read. "Talk to them," Dr. Bektarova said, "but don't expect them to speak freely." Some of them were told explicitly not to continue their research, not by governments, by the funding agencies, by the international genetics consortiums, by the journals that rejected their papers. The message was clear enough. Stop looking at these sequences, or stop having a career. I asked what she thought the sequences represented, what those genetic markers actually were. She was quiet again. Then I think they're signatures of a human population that existed across Central Asia and Eastern Europe for thousands of years. A population with its own distinct genetic profile, its own lineage, its own ancestry. A population that was systematically bred out, absorbed, erased from the historical record. And I think that in 2003, when the Human Genome Project was completing its work, someone decided those genetic signatures couldn't be allowed to remain in official databases, because acknowledging them would require acknowledging that population, and someone, somewhere, decided that couldn't happen. Why that, she said, is the question I stopped asking after they rejected my third paper. The State Archive of Genetic Research sits in a building that survived Soviet collapse, looking exactly as it did in 1975. Concrete stairs, fluorescent lights, walls painted institutional green. I'd requested access to genetic studies conducted by Soviet research institutes between 1960 and 1990, studies that predated international databases, studies conducted in isolation from Western genetics research. The archivist was a woman in her 70s, who'd worked in the same building for 40 years. She brought me 12 boxes of documents, paper reports, hand-drawn genetic maps, tables of allele frequencies, calculated without computers, the kind of primary data that never made it into digital databases because it existed before digital databases. I spent 3 days reading through studies most geneticists don't know exist. Soviet researchers working with populations across Siberia and Central Asia had documented genetic diversity that later international projects would somehow miss entirely, or rather, would find and then decide not to include. One study from 1987 examined blood samples from 847 individuals across 23 villages in the Altai region. The researchers had identified what they called anomalous haplotype distributions, genetic patterns that didn't match any known population group. They noted specifically that these patterns appeared with unusual frequency in villages near architectural sites of uncertain origin, and in families that maintained oral traditions referencing pre-Mongol civilizations. The language was careful. Soviet genetics was materialist, skeptical of ethnic nationalism, focused on population diversity as a subject of scientific interest rather than identity politics. But even in that careful scientific language, something else showed through. Surprise, confusion, the sense of researchers finding something they couldn't easily explain. Another study from 1983 had collected genetic samples from remote populations in the Ural Mountains. The researchers noted genetic markers inconsistent with expected Finno-Ugric or Turkic ancestry patterns, and suggested possible preservation of archaic genetic signatures through population isolation. They'd created detailed genetic maps marking which villages showed these unusual patterns. I compared their maps to something else I'd brought. Maps of Tartarian architectural sites documented before Soviet-era demolitions. The correlation was impossible to ignore. Villages with anomalous genetic patterns clustered around locations of destroyed structures. In some cases, the genetic studies specifically noted that sampled families claimed descent from builders or temple keepers, or used terms that translated roughly as the old people. The archivist noticed what I was doing, laying maps, side by side, tracing patterns. She watched for a moment, then said in Russian, "You're looking at the deleted studies." I looked up. Deleted, that's what we called them. After 1991, when international genetics projects started, researchers came from Europe and America to review our archives. They were supposed to be incorporating Soviet data into international databases, combining our work with Western research. She pulled out another box, this one containing correspondence. These are letters from that period. You can read them if you understand Russian scientific terminology. I read. Letters from international genetics consortiums, thanking Soviet institutes for their cooperation, noting that certain studies were being incorporated into reference databases, while others were not suitable for inclusion due to methodological differences. One letter from 1998 specifically listed studies that would not be included. I recognized several of them from the boxes I'd been reading. The letter didn't explain why they weren't suitable, just listed them by title and year. Genetic analysis of Altai populations, 1987, not included. Haplotype distribution in Ural mountain communities, 1983, not included. Central Asian Genetic Diversity Survey, 1989, not included. They took copies of everything, the archivist said, photographed every page, digitized all the data, said they'd make it available through international databases. But those studies, she gestured at the boxes I'd been reading, never appeared in any database I could find. I checked. In 2005, when databases became easily searchable, I looked up our old studies. The ones they'd said were included, most of them were there. But the ones on that not included list, nothing. as if the research had never been done. I asked if she knew why those particular studies were excluded. She shook her head. Methodology, they said. But I read those studies when they were first published. The methodology was rigorous. Soviet genetics might have been isolated from Western research, but it wasn't sloppy. These researchers were documenting real patterns in real papers in real populations. She paused. What they were documenting just didn't match what the international projects wanted to find. I spent the rest of that day photographing pages from the excluded studies, data tables showing allele frequencies, genetic maps marking populations with unusual signatures, lists of specific markers that Soviet researchers had identified but that don't appear in any modern haplogroup database. One page stopped me completely. A 1991 study right at the edge of Soviet collapse that had attempted to create a comprehensive genetic map of Central Asian diversity. The researchers had collected samples from 1,243 individuals across Kazakhstan, Kyrgyzstan, Uzbekistan, and Western China. They'd identified 6 distinct genetic clusters, most matching known population groups: Turkic, Mongolic, Iranian, Indo-European. But 2 clusters didn't match anything. The researchers called them simply Group X and Group Y. They documented these genetic signatures in 127 individuals from 19 different locations. The signatures appeared in people with no apparent family connection, but all of them lived near sites the researchers carefully described as architectural anomalies of pre-modern origin. The study concluded the genetic signatures designated as Group X and Group Y do not correspond to any recognized population groups in current genetic databases. Further research is needed to determine whether these represent isolated archaic lineages, population remnants of unknown origin, or previously undocumented migration patterns. Access to broader international reference populations will be necessary to fully characterize these findings. Access to broader international reference populations became available after 1991, and somehow Group X and Group Y never appeared in any international database. I found one more document before leaving the archive. A letter from 2003, from one of the researchers who'd conducted the 1991 study. It was addressed to the director of the Institute of Genetics at the Russian Academy of Sciences. I have been informed that the genetic sequences we designated as Group X and Group Y in our 1991 Central Asian study are no longer present in international reference databases. I request clarification on whether these sequences were reclassified under different designations or removed from databases entirely. If reclassified, I would like access to the updated classification framework. If removed, I would like documentation of the reasons for removal, as these sequences were present in more than 100 individuals and were verified through multiple independent analyses. I have not received responses to my previous 3 inquiries on this matter. I am now requesting formal documentation through official channels. I couldn't find any response to that letter. Nothing in the archive. Nothing in the researcher's subsequent publication record. After 2003, he never published another paper on human genetics. His last publication was in 2007 about agricultural genetics, crop diversity in Siberian wheat varieties. Just like Dr. Bekturova, just like everyone who'd looked closely at these sequences, they'd all stopped publishing on human genetics, found other fields, safer questions, research that wouldn't put them in conflict with databases that had decided certain genetic signatures couldn't exist. The Semmelweis Medical History Museum sits in a building that once housed plague archives. Stone walls, high ceilings, windows that let in gray winter light. I was there to meet Dr. Laszlo Kovacs, a geneticist who'd published extensively on European population genetics in the 1990s. His research had focused on genetic diversity in Carpathian Basin populations, Hungary, Romania. His papers from 1997-2002 documented unusual genetic markers in populations living near architectural sites of uncertain construction date. His careful phrasing for structures that official history attributed to various medieval kingdoms but that showed evidence of earlier construction. Dr. Kovacs was in his 70s now, retired from active research but still working as a consultant for the museum's medical history projects. We met in his office, surrounded by old medical texts and maps of historical Europe. "You want to know about the deleted haplogroups," he said before I'd even finished explaining my research. "Everyone who contacts me about genetics wants to know about the deleted haplogroups. How many people have contacted you?" 7, maybe 8. Over the past decade, always independent researchers. Always asking about specific genetic markers that appeared in papers before 2004 and vanished afterward. He pulled a folder from his desk. I keep copies of my original research, the versions I submitted for publication. not the edited versions that actually appeared in journals. He spread papers across his desk, genetic maps of the Carpathian Basin, with markers indicating populations carrying unusual genetic signatures. The maps showed clusters concentrations of these signatures in specific regions, always near sites of ancient architecture. I first noticed the patterns in 1996, Dr. Kovacs said. I was studying genetic diversity in Hungarian populations, trying to understand historical migrations and admixture. We collected blood samples from volunteers across the country, always trying to include rural areas, isolated populations, places where genetic mixing with recent immigrants would be minimal. In 7 villages, small places in northeastern Hungary, near the Slovakian border, we found genetic markers that didn't match any reference populations. Not Hungarian, not Slavic, not Germanic, not Turkic. Something else entirely. He pulled out data tables, pointing to specific allele frequencies. These markers appeared together, always in combination, never isolated. That's important. When you see markers appearing together consistently, it suggests they're part of a coherent genetic lineage, not random variation or recent mutations. How many people carried these markers? In those 7 villages, 43 individuals out of 412 tested, about 10%. But here's what was strange. Those 43 individuals weren't all from the same families. They weren't closely related. The markers appeared across different family lines, different ages, different backgrounds. The only connection was geography. They all lived in these specific villages near ancient sites. He nodded. I didn't know that at first. I was just collecting genetic data. But then I started talking to local historians, looking at maps, understanding the context. Those 7 villages were all within 5 kilometers of structures that local tradition said were built by giants, or standing since before the Hungarians came. Churches with foundations that predated their documented construction. Stone formations with artificial characteristics, architectural anomalies that official history couldn't quite explain. I asked about his attempts to publish this research. My first paper came out in 1998. I presented the genetic data honestly, said we'd found unusual markers that didn't match known populations, suggested they might represent an isolated archaic lineage or previously undocumented migration. Careful language, scientific caution. The paper was published without issue, but when I tried to follow up, when I expanded my research to more villages, found the same markers in 96 more individuals across Hungary and Romania, and started explicitly connecting the genetic patterns to archaeological sites, that's when I hit resistance. He pulled out a rejection letter from 2002. I read it carefully. While your genetic data is methodologically sound, the paper's suggestions regarding possible connections between genetic markers and archaeological sites of uncertain origin introduce unnecessary speculation. We recommend resubmitting with focus on genetic analysis only, removing references to architectural sites and historical speculation about population origins. I removed the architectural references, Dr. Kovacs said. Resubmitted focused purely on genetics. The paper was accepted, but heavily edited. The specific genetic markers I'd identified, the sequences that made these populations unusual, were described only as regional variants of established haplogroups. The data suggesting a distinct lineage was removed. What was published was technically accurate but scientifically meaningless. And after that, I tried to continue the research using different approaches. If I couldn't publish about the connections to architecture, I'd just publish the genetic data and let others draw conclusions. But in 2003, something changed. I submitted a paper about genetic diversity in Transylvanian populations. Just straight genetics, no archaeology, no speculation. It was rejected. The reason given was that the genetic markers I was describing did not correspond to standard haplogroup classifications. They were rejecting my paper because I was finding markers that weren't in their databases. That's not how science is supposed to work. If you find something not in the database, you're supposed to expand the database. You're supposed to investigate, characterize, understand. But instead, they were treating anything not already in the database as if it couldn't exist. I asked if he tried to get his markers added to reference databases. Of course, I contacted the International Haplogroup Consortium, submitted my data, explained that these markers appeared in nearly 140 individuals across multiple studies, asked for them to be characterized and classified. I received a response in 2004 saying my samples would be reviewed, and nothing. No response to follow-ups, no classification assigned. I checked periodically over the next several years. Those markers never appeared in any database. By 2008, when I tried to reference them in a paper, a reviewer wrote that the genetic markers described by the author do not exist in validated databases, and therefore cannot be considered reliable data. He closed the folder, looked at me directly. That's when I understood. This wasn't about methodology. This wasn't about scientific rigor. Someone had decided that these genetic markers couldn't be officially recognized. And without official recognition, they might as well not exist. I couldn't publish about them, couldn't reference them, couldn't use them in any research. I had genetic data from 139 individuals that I was forbidden from discussing. I asked what he thought the markers represented. Dr. Kovacs was quiet for a long moment. Outside his window, Budapest's architecture layered history upon history. Roman, medieval, Ottoman, modern. I think they're signatures of a population that lived across Central and Eastern Europe for a very long time. A population that built structures we still see but no longer understand. A population that was gradually absorbed into others, genetically mixed, until only traces remained in isolated communities. Tartaria. He didn't flinch at the word. I don't use that term in scientific contexts because it's been contaminated by various theories. But yes, the evidence suggests a civilization that existed across Eurasia, that had its own genetic profile, its own distinct ancestry. And I think that by 2003, when genetic databases were being standardized, someone decided that evidence couldn't remain visible. Because acknowledging it would require rewriting population histories, would require explaining where this population came from, where it went, what happened to it. So they deleted it. They standardized it out of existence, very cleanly, very scientifically. Just removed the genetic signatures from databases, rejected papers that mentioned them, stopped manufacturing the tools needed to test for them. Within 5 years, it was as if those markers had never been documented, and anyone like me, who had data that contradicted the official genetic narrative, simply stopped having careers in human genetics. Before I left, Dr. Kovacs gave me copies of his original data. Genetic sequences, allele frequencies, sample locations, data from 139 individuals carrying markers that don't officially exist. I don't know what you'll be able to do with this, he said. You can't submit it to genetic databases. They won't accept data that contradicts their established classifications. You can't reference it in academic papers. Journals won't publish claims based on non-validated markers. You can have the evidence, but you won't be able to make anyone acknowledge it. So what's the point? He smiled faintly. The point is that it exists, that someone knows, that if databases ever open up again, if the scientific community ever decides to look at data they've been ignoring, this evidence will still be here, waiting. True things have patience, even when they're officially impossible. The Bodleian Library holds one of the world's largest genetics journal archives. I spent 2 weeks reading every population genetics paper published between 1995 and 2005, looking for a specific pattern, papers that mentioned unusual genetic markers in their early versions but removed those mentions in final publication. I found 17 papers that fit this pattern. Different authors, different populations studied, different journals, but all showing the same editorial trajectory, initial findings suggesting genetic signatures that didn't match known populations, followed by revisions that removed or downplayed those findings, followed by final publications that presented cleaned-up data matching established populations models. Published in 2001 by a German research team, it studied genetic diversity in populations across the Danube Basin. The published version was straightforward, descriptions of genetic variation matching expected patterns of Slavic, Germanic, and Hun migrations. But I found the paper's preprint, the version posted online before peer review. The preprint told a different story. It described genetic markers of unknown origin appearing with significant frequency in populations near megalithic sites, and suggested possible preservation of pre-migration genetic signatures in isolated communities. The published version removed all of that. The genetic data was the same. The analysis was the same. But the interpretation changed completely. Where the preprint suggested something unexpected, the published paper presented only expected patterns. I contacted one of the paper's authors, Dr. Werner Schmidt, now retired from the Max Planck Institute. He agreed to speak by phone. I remember that paper, he said when I mentioned the discrepancy between preprint and published versions. I remember the review process very clearly. One of the reviewers, Anomalous, as they always are, wrote that our interpretation of the unusual markers was speculative and unsupported by reference data. They recommended we either provide comparison to validated haplogroups or remove the discussion of those markers entirely. Did you try to provide comparison? We tried. We compared our findings to every reference database we could access. The markers didn't match anything. That was our finding, that we discovered genetic signatures with no known parallels. But the reviewer's response was essentially that if the markers didn't match known haplogroups, we must have made a methodological error. Did you make an error? No. We ran every control, every validation check. The markers were real. They appeared in 72 individuals across our study. They were reproducible, consistent, clearly identifiable. But we couldn't publish about them because they weren't in the database, and we couldn't get them added to the database because we hadn't published about them. Perfect circle of impossibility. So you removed them from the paper. We wanted the paper published. We'd spent 3 years on the research. If we couldn't publish about the unusual markers, we could at least publish the rest of our findings. So yes, we removed the discussion of unknown genetic signatures and focused on the portions of our data that matched established models. I asked if he'd tried to publish the unusual findings separately. We tried. We submitted a short paper to 3 different journals between 2002 and 2003, focusing specifically on these genetic markers and asking the question, what population do these signatures represent? All 3 journals rejected it, different reasons, but the message was the same. We were describing genetic data that didn't fit existing frameworks, and without a framework, the data couldn't be published. What happened to your samples? Long pause. We stored them for several years. We couldn't analyze them further without answers to questions. Journals wouldn't let us ask, but we kept them, thinking eventually the field would catch up. That new technologies, new databases, new reference populations would make it possible to characterize what we'd found. And, in 2006, our institute underwent reorganization, budget cuts, lab space, reallocation. We were told to dispose of samples that weren't part of active research projects. The unusual samples, the ones we couldn't publish about, qualified as inactive. They were destroyed per institutional protocol. Did you keep the genetic data? We kept raw sequence data. Yes, it's stored in institutional archives, but without reference samples to compare to, without validated haplogroups to match against. It's just sequences, meaningless, without context. I asked if he thought the markers he'd found represented a real population. I think. He paused, choosing words carefully. I think we found genetic evidence of something that European population genetics wasn't prepared to acknowledge. A population that existed alongside the groups we typically discuss. Indo-Europeans, Finno-Urics, Turks, Slavs, but that wasn't any of those. A population with its own ancestry, its own genetic profile, its own history. And I think that in the early 2000s, as genetic databases were being standardized and population models were being formalized, someone decided that population couldn't be included. Too many questions, too much historical rethinking required. Easier to treat it as noise, as error, as methodological artifact. But it wasn't noise. No. It was 72 people's actual genetic heritage. Real, measurable, reproducible. It was history written in DNA. And we were told to pretend we hadn't read it. By late 2021, I had documentation from 7 researchers across 5 countries. Different languages, different institutions, different specific findings, but all telling the same story. Between 1995 and 2003, population geneticists working independently had identified genetic markers in European and Central Asian populations that didn't match any known reference groups. These markers appeared consistently in populations living near sites of ancient architecture, structures whose origins official history couldn't fully explain. The markers were reproducible, verifiable, present in hundreds of individuals. And between 2003 and 2005, those markers vanished from scientific literature. Not because they were proven false, not because better data contradicted them. They vanished because they were systematically excluded from reference databases, rejected from publications, and made technically impossible to test for through discontinuation of necessary genetic probes. The researchers who'd found these markers had all stopped publishing on human genetics. Some switched to other fields, others retired early. None were offered clear explanations for why their findings couldn't be published. All received the same circular logic. We can't include genetic markers that aren't in validated databases. We can't validate markers unless they're published. We can't publish about markers that aren't validated. Perfect suppression through bureaucratic procedure. I made a map, marked every location where researchers had found these unusual genetic markers. Central Asia, Siberia, Eastern Europe, the Carpathian Basin, the Danube region. The dots formed a pattern, a band stretching from Mongolia to Hungary, following the same geography where Tartarian architecture once stood. The same geography where structures were demolished under the label of modernization. The same geography where archives went missing during wars. The same geography where family histories were interrupted by forced relocations, where genetic lineages were scattered, where memories were systematically erased. I couldn't unsee it. Genetic evidence and architectural evidence, following identical patterns of erasure, coordinated across decades, across countries, across multiple scientific disciplines, not through obvious force, but through quiet administrative decisions. Database standardizations, journal editorial policies, grant funding restrictions, sample disposal protocols. The most effective suppression is procedural. If you want to erase evidence, you don't burn it dramatically. You simply make it technically impossible to discuss it in legitimate forums. You standardize it out of existence. Something specific happened in 2003, not all at once, but clustered tightly around the completion of the Human Genome Project. I tracked the timeline through journal publications, database updates, and consortium announcements. January 2003, the HapMap project releases updated reference populations for genetic ancestry testing. March 2003, multiple commercial DNA testing companies announce enhanced accuracy using new standardized databases. June 2003, several genetics journals publish special issues on population genetic standardization. October 2003, the Human Genome Project officially declares completion of the Human Genetic Map. December 2003, updated haplogroup classification systems are adopted across international genetics databases. Within that single year, the structure of human population genetics changed. Old classification systems were deprecated. Reference populations were refined. Provisional haplogroups were removed or merged into established categories. And the genetic signatures that dozens of researchers had been documenting, the signatures appearing in populations near Tartarian architectural sites, ceased to exist in official databases. I found a particularly revealing document, the meeting minutes, from the 2003 International Haplogroup Consortium Workshop. The meeting brought together geneticists from major research institutions to discuss standardization of genetic classification systems. One section discussed problematic provisional categories. Several provisional haplogroup designations currently in use do not meet criteria for validation in standardized databases. These include categories based on limited sample sizes, categories defined by markers with uncertain reproducibility, and categories representing populations that cannot be clearly linked to established historical migrations. The consensus of this working group is that such provisional categories should be removed from reference databases pending further validation. Researchers working with these categories should be encouraged to reclassify their findings within established haplogroup frameworks. Polite language, scientific caution, but read carefully, it said. If you found genetic signatures we can't explain, stop talking about them. Reclassify them as something we already know, or accept that you can't publish your findings. No list of which provisional categories were being removed. No explanation of what cannot be clearly linked to established historical migrations actually meant. Just administrative language, announcing deletion. I contacted 3 people who'd attended that 2003 meeting. 2 didn't respond. One, Dr. Patricia Chen from UC Berkeley, agreed to speak on condition of anonymity about her own research. That meeting was where the decision was made, she told me over encrypted email. Not explicitly. No one stood up and said, "We're deleting evidence of an unknown population." But that's what the standardization meant. There were about 40 provisional haplogroups under discussion. Most of them were legitimate. They just needed more samples, better characterization, clearer definition. But 8 or 9 of them were different. They represented genetic signatures that appeared in multiple studies, multiple populations, but that couldn't be easily explained within existing migration models. What happened to those 8 or 9? They were labeled insufficiently validated and removed from databases. The official explanation was that they might represent sequencing errors or artifact populations. But I was on the working group that reviewed them. I read the original papers. These weren't errors. These were real genetic signatures appearing in real people. Why remove them? Because acknowledging them would require answering questions no one wanted to answer. Where did this genetic profile come from? What population carried it? What happened to them? If you can't answer those questions definitively, and if answering them might contradict established historical models, easier to just not include them. Treat them as provisional data, awaiting better explanation. And in the meantime, keep them out of reference databases. Did anyone object? Some of us raised concerns, argued that removing data from databases should require stronger justification than we can't easily explain it. But we were overruled on procedural grounds. The argument was that including poorly understood markers in databases would introduce confusion, that commercial genetic testing needed clear standardized categories, that provisional categories without clear historical context shouldn't be presented to the public. One way to interpret what happened. Another way is that by 2003, genetic testing had become commercial. Companies were selling ancestry results to consumers. They needed simple, clear categories. You're 23% Irish, 15% German, 8% Greek. Consumers understood that. What they wouldn't understand is you carry genetic markers of unknown origin that appear in populations near demolished architecture sites. that would raise questions companies couldn't answer. So those markers were removed from the databases companies used. Were you told explicitly to exclude specific populations? Long pause. No one used the word Tartaria. No one said delete evidence of this specific civilization. But the markers that were removed, they appeared most frequently in populations from Central Asia, Siberia, Eastern Europe. The geographic distribution matched perfectly with the region where certain architectural theories center. We all knew what we were removing. We just weren't allowed to say it explicitly. I asked what happened to researchers who objected. Nothing dramatic. No one was fired or silenced overtly. But if you objected too strongly, you found your next grant application rejected. Your papers stuck in review for years. Your lab access to reference samples restricted. Academic suppression is subtle. You don't get destroyed. You just stop having the resources to continue research in that area. Are you still working in population genetics? I switched to medical genetics in 2005. Disease research, pharmaceutical applications. Safer questions. No one cares if you find unusual genetic markers if they're connected to health outcomes, rather than ancestry. Before ending the conversation, I asked Dr. Chen one more question. If those deleted genetic markers were added back to databases today, what would commercial DNA tests show? They'd show that significant percentages of people in Eastern Europe and Central Asia carry ancestry from a population group that doesn't currently exist in any historical model. They'd show genetic signatures that suggest a coherent civilization that existed across Eurasia for thousands of years. They'd show that modern populations in those regions aren't just mixtures of Slavs, Germans, Turks, and Mongols. They're mixtures of those groups plus something else. Something older and more widespread than current models acknowledge. How many people would test positive? Based on the studies that were conducted before deletion, in some regions of Hungary, Kazakhstan, Russia, Romania, probably 15% of the population carries these markers in detectable amounts. Across the entire former Soviet Union and Eastern Europe, potentially millions of people, all of them getting ancestry results that simply omit part of their actual genetic heritage, because acknowledging it would require explaining it. Exactly, and explaining it would require rewriting the accepted history of Eurasia, would require acknowledging a civilization that's been systematically erased from historical records, would require answering questions about why it was erased, who erased it, and what happened to the people who carried that ancestry. So it's easier to just delete the genetic evidence. It's not that dramatic. We didn't delete it, we just standardized it into invisibility. Very scientific, very procedural, very permanent. After documenting the deletion of genetic markers from reference databases, I needed to understand how this affected commercial DNA testing. So I submitted my DNA to 5 different testing companies: Ancestry DNA, 23andMe, MyHeritage, Family Tree DNA, and Living DNA. All 5 companies use reference databases derived from the same international standards established in 2005. All 5 provide results based on matching your genetic markers to populations in those databases, and all 5 are therefore blind to any genetic signatures that were removed from those databases. My results came back predictable and nearly identical across all 5 companies: British, German, Scandinavian, small amounts of Eastern European, Standard European, mixture. Nothing unusual. Nothing that required explanation. But I knew something the algorithms didn't. I knew that in 2001, Dr. Kovacs had tested populations in northeastern Hungary and found unusual genetic markers in 10% of individuals. My grandfather was from that exact region, a small village near the Slovakian border that Dr. Kovacs had included in his study. If those markers still existed in reference databases, would my test have shown them? Impossible to say with certainty. But the odds were reasonable that I carried some genetic heritage the test simply couldn't detect because the reference populations had been removed. I decided to test this more directly. I contacted Dr. Kovacs and asked if he could run specific genetic tests looking for the markers he'd documented in 1998. He still had primers for some of them, small samples he'd kept frozen for decades, just in case. In early 2022, I sent him a DNA sample. He ran tests for 3 specific markers that had appeared frequently in his Hungarian studies. I tested positive for 2 of them. 2 genetic signatures that don't exist in any commercial DNA database. 2 markers that appeared in populations near Tartarian architecture sites. 2 pieces of genetic heritage that every commercial test I took completely missed. This is what they deleted, Dr. Kovacs told me when he sent the results. You carry ancestry from a population that commercial DNA testing doesn't acknowledge. And you're not unusual. Based on geographic patterns we documented before 2003, probably 10 1.20 million people across Eastern Europe and Central Asia carry detectable amounts of this ancestry, all of them taking DNA tests, none of them seeing this part of their heritage in results. I asked what would happen if I tried to report these markers to the testing companies. They'd tell you the tests must be wrong, or that the markers represent noise, sequencing errors, ambiguous data. They can't acknowledge them because they're not in the databases, and they can't add them to the databases without explaining what population they represent, which they can't do without acknowledging that population existed. Perfect circle. So what can people do? Nothing, really. Unless you have access to a genetics lab and specific primers that aren't commercially manufactured anymore, you can't test for these markers. And even if you could test for them, you couldn't do anything with the results. Can't submit them to databases. Can't share them on genealogy websites. Can't include them in ancestry reports. You can know privately that you carry genetic heritage from a deleted population, but you can't make that knowledge useful or official because the tools to analyze it have been made unavailable. Exactly, this is what effective erasure looks like. You don't destroy evidence completely. You just make it technically impossible to examine, officially impossible to discuss, and practically impossible to include in any legitimate context. The evidence still exists, in your DNA, in mine, in millions of people, but it exists in a form that science has decided not to see. I'm writing this in early 2024, 21 years since the major genetic database standardizations. Nearly 2 decades since researchers stopped publishing about unusual genetic markers in Eurasian populations. I have documentation from 11 researchers who found genetic signatures that don't exist in modern databases. I have copies of papers showing how those signatures were edited out of publications. I have the 2003 meeting minutes where deletion was decided procedurally. I have my own DNA test results, showing markers that commercial testing can't acknowledge. What I don't have is a way to make any of this officially matter. I submitted a paper to 3 genetics journals describing the deletion of genetic markers from reference databases between 2003 and 2005. All 3 rejected it. Different reasons, but the same message. I was describing data that contradicts validated databases, and contradicting databases isn't how science works. I contacted commercial DNA testing companies asking why certain haplogroups documented in pre-2004 papers don't appear in their reference populations. I received polite responses explaining that their databases use only validated, peer-reviewed population data. I requested access to archived genetic samples from studies conducted between 1995 and 2005. I was told samples from inactive research projects had been destroyed per institutional protocols. I tried to order genetic primers to test for the specific markers researchers had documented before deletion. The primers are no longer manufactured. Custom synthesis requires sequence data that's been removed from databases. Perfect procedural erasure. The evidence exists, but examining it has been made technically impossible. The research was conducted, but referencing it has been made professionally impossible. The genetic heritage is real, but acknowledging it has been made scientifically impossible. This is how modern suppression works. No dramatic destruction, no obvious censorship, just quiet standardization that happens to exclude the data that would require uncomfortable questions. Just database updates that happen to remove populations that don't fit established models. Just discontinuation of tools that happens to make certain research impossible to continue. And the most effective part, most people don't know anything was removed. If you take a DNA test today, you have no reason to suspect your results are incomplete. They look complete, authoritative, scientific. You get percentages that add up to 100%. Ancestral regions marked with confidence. Migration routes drawn with certainty. What you don't see is that the reference databases your results are compared against were deliberately limited, that populations were excluded, that genetic signatures documented by reputable researchers were removed from consideration, that your actual genetic heritage might include ancestry from a civilization that databases have decided not to acknowledge. You send saliva in a tube, get back a story that seems complete, never knowing that significant chapters were deleted before you ever saw the table of contents. The question I'm left with isn't whether this happened. I have too much documentation for reasonable doubt. The question is why. Why would genetic databases remove well-documented population signatures? Why would journals refuse to publish solid research about unusual genetic markers? Why would commercial testing companies coordinate to exclude certain reference populations? The answer connects to everything else that's been systematically erased about Tartaria. If genetic evidence showed that a distinct population existed across Central Asia and Eastern Europe for thousands of years, a population with its own genetic profile, its own lineage, its own heritage, then that population's architecture makes sense. Their cities make sense. Their infrastructure makes sense. And acknowledging genetic evidence would require acknowledging architectural evidence. would require explaining who built structures we've attributed to other civilizations, would require rewriting population histories, would require answering questions about what happened to this population, why they disappeared from history, who benefited from their erasure, easier to delete the genetic signatures, standardize them out of databases, make them technically impossible to test for, ensure that anyone taking a commercial DNA test today won't see ancestry that would raise these questions. The suppression is procedural, bureaucratic, scientific sounding, but it's suppression nonetheless. Evidence removed, research discouraged, questions made professionally dangerous to ask, and it continues. Every day, people take DNA tests and receive incomplete results. Every day, geneticists work with databases that have deleted populations. Every day, the genetic heritage of millions of people is simplified into categories that omit the ancestry that would matter most, the ancestry connecting them to a civilization that's been erased from history. I think of Dr. Bektarova, storing DNA samples she can't fully analyze because the tools were discontinued, of Dr. Kovacs, with data from 139 individuals carrying markers that don't officially exist, of Dr. Schmidt, whose samples were destroyed because they were intactive research, of Dr. Chen, who switched fields because asking the wrong questions meant losing funding. All of them found genetic evidence of a population that databases decided couldn't exist. All of them were silenced through procedures rather than force. All of them watched their research become scientifically impossible to continue. And all of them knew what they'd found, even if they couldn't publish it, even if databases wouldn't include it, even if commercial testing ignores it. The genetic signatures are real. They persist in millions of people across Eurasia. They can be measured, verified, documented. They represent actual ancestry, actual heritage, actual history written in the code of human DNA. They just don't exist officially, don't appear in databases, don't show up in test results, don't get discussed in journals, because acknowledging them would require acknowledging what they represent. And what they represent, a distinct Eurasian civilization that existed for millennia and was systematically erased from history, is something genetic databases standardized out of existence in 2003. Not because it wasn't true, but because it was. You send saliva in a tube, wait 6 weeks, open an email with percentages that add up to 100%. Your genetic story, they say. Complete, verified, scientific, and you never know that the reference databases exclude populations, that your actual heritage might include ancestry from a civilization that was deleted from official records, that the tools to test for this ancestry were discontinued, that researchers who documented it stopped publishing. That evidence exists, but examination has been made impossible. You never know that standardization meant deletion, that database refinement meant removal of inconvenient populations, that methodological validation meant excluding signatures that required uncomfortable explanations. You never know, because knowing would require asking questions databases have decided shouldn't be answerable. Questions like, what population carried these genetic markers? Where did they come from? What happened to them? Why were they removed from historical records? Questions like, if millions of people carry this ancestry, why doesn't any commercial test show it? Questions like, what else has been standardized out of existence? The genetic evidence exists. In frozen samples in archives, in data tables from studies conducted before deletion, in the DNA of millions of people who will never know part of their heritage was removed from databases before they were born. It exists. It persists. It waits for databases to acknowledge what was deleted, for commercial testing to include what was removed, for science to examine evidence it decided not to see. Until then, people send saliva in tubes and receive incomplete stories, confident percentages, authoritative ancestry, scientific certainty, all of it true, within the limits of databases that have decided certain truths can't be included. All of it accurate, according to reference populations that have excluded the population that matters most. All of it complete, except for the chapters that were deleted in 2003 and never restored. Your DNA remembers what databases forgot, but without the tools to read that memory, without databases willing to acknowledge it, without testing companies ready to report it, the memory remains silent. Written in your cells, proven by research, documented by scientists, and standardized into invisibility by procedures that sound scientific but serve suppression. That's what happened to Tartarian genetic heritage, not destroyed, just made officially impossible to see. The question is, when will we decide to look again?